Welcome, future medical professionals, to the Deep Dive. Today, we're diving into something, well, truly incredible. A hidden world right inside you that's reshaping how we think about health. Expert, this idea that we are more microbe than human. It's kind of mind-bending, isn't it? Can you maybe set the stage for us? What exactly is this internal microbial city?
It absolutely is mind-bending, and it's a fantastic place to start because that phrase, it's more than just catchy. It signals a really profound shift in perspective. Reliable medical sources are showing us that the human body isn't just, our cells. It's this complex, bustling ecosystem, a microbial city teeming with microorganisms. We call this community the microbiota.
Okay, microbiome.
And they're not just hanging out. They form what we're now calling a virtual organ. It has these extensive, well-documented roles crucial for our health, even our survival.
Yeah.
So this isn't just abstract biology for you. It's a fundamental part of human physiology that's, well, rewriting medical textbook.
A virtual organ.
Exactly. And the scale is immense. Think about roughly 100 trillion microbes. They're on your skin, in your mouth, but the vast majority residing in your gastrointestinal tract. Their collective impact is huge.
Right, that really changes how we need to think about our future patients.
Yeah.
So our mission today, especially for you listening, our future medical colleagues, is to really get a solid foundation here. We want to unpack this hidden world, understand its impact, how we get it, what happens when it goes wrong.
Dysbiosis.
Dysbiosis, yeah.
And crucially, what we can actually do about it in the clinic. This stuff is going to pop up everywhere in your studies. So, okay, this microbial city, let's break it down. For someone just starting out, what exactly is the microbiota and who are these invisible roommates?
Right. So at its heart, the microbiota is simply a community of microorganisms. And they inhabit a specific environment, in this case, us, the human body. It's usually a mutualistic relationship. They get a place to live, and we get, well, a lot of benefits. They're not freeloaders.
Okay, so it's a community. But who's in it? Just bacteria. Ugh, good question. Bacteria are definitely the most famous residents, the workhorses. Incredibly diverse. But no, it's much more varied. We also have viruses living in us, mostly things called bacteriophages. Think of them like tiny bacterial police that specifically infect bacteria, helping keep populations in check.
Bacterial police, sounds
Yeah.
like that.
Then you've got archaea, like Methanobrevibacter. It actually helps break down nutrients by using hydrogen gas produced by other microbes.
Right.
We also have fungi, like Candida species, which you'll definitely encounter, and even some protozoa. It's a whole multi-kingdom ecosystem in there.
That's way more diverse than I think most people realize. So for our listeners, the medical students trying to anchor this, especially in the gut, which you said is the main hub, what are the big bacterial groups, the key players?
Excellent question. You definitely want to focus on the dominant bacterial phyla in the gut. To make it easier to remember, let's use an acronym, FBAP.
FBAP.
Yeah, FBAP, it stands for Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria. These four are really the major players, and their relative balance tells us a lot about gut health.
Okay, FBAP. Can you give us quick examples for each, just names we might start recognizing?
Sure. So for Firmicutes, think Lactobacillus, which you know from probiotics, Clostridium, some good, some bad, like C. diff and Streptococcus. For Bacteroidetes, the big one is Bacteroids. They're great at breaking down plant fibers. For Actinobacteria, Bifidobacterium is key, really important in healthy infants. And finally, Proteobacteria. This includes things like E. coli. Yes, the E. coli and Helicobacter pylori. Now, some proteobacteria can cause trouble, but many are normal residents. An overgrowth of this group, though, often signals something's off balance.
Got it. FBAP, Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria. That's helpful. Okay, so here's where it gets really interesting for me. We know who they are now, these invisible roommates. But what are these mighty miniatures actually doing for us? What makes them this virtual organ?
Right, this is the core of it. They're active collaborators in our physiology. It's not just us doing everything. Think of them as tiny nutrient factories, for one. They break down complex carbohydrates, things our own enzymes can't touch. Fiber, for instance. And doing that produces beneficial short-chain fatty acids, SCFAs. You'll hear a lot about butyrate, propionate, acetate. These SCFAs, they feed our colon cells, but also influence things systemically.
Okay, nutrient factories. What else?
Oh, a lot. They're crucial for regulating our immune system. They literally help train it from birth, influencing how immune cells develop. They help maintain the integrity of our gut barrier, that single layer of cells lining the gut. Think of it as our internal security wall, keeping bad stuff out.
The security wall.
Exactly. They ferment fibers, synthesize essential vitamins like vitamin K, some B vitamins, metabolize plant compounds. They also protect us against pathogens. They compete for space and food. And some even produce antimicrobial substances, bacteriocins, that target harmful bacteria.
Wow.
And get this, through the gut-brain axis, research suggests they even play a role in our mental health and regulating metabolism, body weight. It's extensive.
It really is a whole other layer of complexity to the human body, which leads to a really basic question. If they're so critical, how do we even get them in the first place? Like, what's the microbial blueprint for a human? How does that initial setup happen?
That's the million-dollar question, isn't it? How does this all start? There are basically two main ways we acquire our microbiota: vertical and horizontal transmission.
Vertical and horizontal.
Vertical is the initial seeding from parent to offspring. Primarily, this happens during childbirth. Vaginal delivery exposes the baby to the mother's vaginal and fecal microbes, which is a really important first inoculation. Breastfeeding continues this, providing more microbes and also special sugars that feed beneficial bacteria like Bifidobacterium.
Right, so that initial exposure is key. Can you give us a clinical example of why understanding that vertical transmission is so important?
Absolutely. Let's take Group B Streptococcus, GBS streptococcus echolactiae. It's a common bug, colonizes the GI and genital tracts in many healthy adults, often without symptoms. But during childbirth, it can be transferred to the baby either as it passes through the birth canal or sometimes even before birth if it ascends. And this matters clinically because GBS is a major cause of really serious infections in newborns under 3 months. Think sepsis, meningitis, pneumonia. We've known this since the 70s, which is why pregnant women are screened for GBS. Understanding that vertical route is critical for prevention.
That makes perfect sense and directly impacts patient care. But hang on, you mentioned vaginal delivery specifically. What about C-sections? Does that mean those babies are sort of starting behind? Is it that simple?
That's a really important nuance, and no, it's not quite that simple. Studies do show babies born via C-section tend to have an initial microbiota that looks more like skin microbes, from the mother in the hospital environment rather than the vaginal microbes. But the long-term health implications, that's still being heavily researched. It's not like a guaranteed disadvantage. Many other factors come into play later: diet, environment. But understanding these early differences, it helps us think about potential ways to support microbial development in those infants. So it's a factor, definitely, but not the whole story.
Okay, that's a more balanced view. So that's vertical transmission. What about horizontal?
Horizontal transmission is everything else acquiring microbes from the environment after birth. So interacting with family members, pets, playing outside, just living life. And a huge factor here is diet. Diet is probably the major microbial architect throughout our lives. What you eat constantly shapes and reshapes who lives in your gut.
The microbial architect. I like that.
Yeah.
Okay, so we build this blueprint, it evolves. But what happens when this microbial garden, as you called it, goes wild? When the balance is off? What's the clinical picture there?
Right. When things get out of balance, the term we use is dysbiosis. It basically means an alteration in the composition of the gut microbiota. This could be a loss of beneficial microbes, an overgrowth of potentially harmful ones, or just a general loss of diversity. And importantly, dysbiosis can be both a cause of disease and a consequence of it. It's often a vicious cycle.
A vicious cycle. And you mentioned earlier, this isn't just about gut problems. So if dysbiosis sets in, what kinds of conditions are we talking about? What might we see in patients, even outside the GI tract?
You're right. The impact is broad, really systemic. Within the gut, dysbiosis is strongly linked to things like IBS, irritable bowel syndrome, also inflammatory bowel diseases, IBD, so Crohn's and ulcerative colitis, celiac disease too, and even colorectal cancer risk.
Okay, those make intuitive sense. Yeah. But systemically?
Well, the connections are getting stronger all the time. We see links to metabolic syndrome, that cluster of conditions like obesity, type 2 diabetes, fatty liver disease, and perhaps most surprisingly, there are links being explored with neurological conditions, things like autism spectrum disorders and even neurodegenerative diseases like Alzheimer's and Parkinson's. It really underscores that gut-brain connection and how far-reaching the microbiota's influence is. It's not just digestion, far from it.
That's truly profound. It changes the lens on so many diseases. Which brings up a really practical question for future clinicians listening. If the balance is off, what can we do? How do we intervene? And, what about the things we do, like prescribing antibiotics? How does that impact this delicate garden?
That's a critical point about antibiotics. They're lifesavers, no question, absolutely essential. But they do alter the microbiome structure. They're often broad spectrum, so they hit the good guys along with the bad. Now, the overall structure usually bounces back, eventually. But here's the crucial part. Research shows the genomic structure, the specific genetic capabilities of the community, might not return to exactly what it was before. There can be lasting, subtle shifts. It highlights the need for judicious antibiotic use.
Right, a lasting impact, potentially. So besides being careful with antibiotics, what are the positive interventions?
We have several symbiotic interventions, ways to try and support a healthy balance. You've likely heard of probiotics. Think of these as microbial reinforcements. Live microorganisms, specific bacteria or yeasts that, when you take them in adequate amounts, are intended to have a health benefit.
Like in yogurt or supplements?
Exactly. Then there are prebiotics. These are like microbial fertilizer. They're usually types of fiber or non-digestible compounds that selectively feed the beneficial microbes already living in your gut. Help the good guys flourish. And sometimes you see symbiotics, which combine both probiotics and prebiotics.
Reinforcements and fertilizer.
Yeah.
Okay. What about more drastic measures?
Well, the most dramatic is probably fecal microbiota transplantation, or FMT. You can think of this as the ultimate microbial reset button.
Right, transferring stool from a healthy donor, it sounds intense.
It does. When I first heard about it, I admit it sounded pretty wild. But for treating recurrent Clostridium difficile infection C. diff, it has a remarkable success rate, around 90%. It shows just how powerful resetting that microbial environment can be. And it's not actually brand new. Conceptually, there are records of similar practices centuries ago, even in veterinary medicine.
A reset button. Incredible. So we're learning to manage the ecosystem. But sometimes the challenge isn't just balance, it's structure, right? These really tough microbial communities. We definitely need to talk about biofilms. What are these bacterial fortresses?
Yes, biofilms are a huge challenge clinically. They are essentially organized communities of microbes. They stick to surfaces, could be our own tissues, like teeth-causing plaque or often medical devices, and they surround themselves with this protective slime layer, this extracellular matrix they produce. It shields them.
So like a fortress wall.
Exactly. And inside this fortress, things happen. Microbes can exchange genes more easily, including resistance genes. The NIH estimates that over 80% of chronic microbial infections are associated with biofilms. 80%.
Wow, that's huge.
It is. And critically, bacteria within a biofilm are significantly more resistant to antibiotics than their free-floating counterparts, sometimes hundreds or thousands of times more resistant. So think about dental plaque, like I mentioned, chronic lung infections in cystic fibrosis patients, and a really big one, infections on medical implants, catheters, artificial joints, heart valves. These are notoriously difficult to treat, often requiring removal of the device.
That makes sense why they're such a problem. Increased resistance, hard to reach.
Precisely. So thinking clinically again, considering these fortresses and their drug resistance, what does that mean for treatment? It forces us to think beyond standard antibiotics, look at prevention, ways to disrupt the biofilm itself. It's a major area of research and a practical challenge you will face.
Okay, let's try and unpack all of this as we wrap up. We've journeyed through this amazing microbial city inside us. We saw it's not just microbes, but a vital virtual organ. We learned how we get our initial microbial blueprint and how diet acts as the architect. We explored dysbiosis, the unbalanced garden, and its links to so many diseases. And we look at interventions, the reinforcements, fertilizer, the reset button, and the challenge of those tough bacterial fortresses, the biofilms. For you listening, our future colleagues, the importance here is massive. This isn't just some niche topic. It's fundamental to understanding health, disease, immunity, metabolism, pretty much everything.
Absolutely. And maybe a final thought to leave you with. As you start your medical journey, always remember that every single patient carries this unique, dynamic internal world. Understanding that host microbe interaction isn't just about fighting infections. It's about seeing the whole picture of health. It's about recognizing these powerful, unseen forces within us. And honestly, the more we learn, the more doors open for truly personalized medicine, for new ways to diagnose and treat. So stay curious. Keep observing. This microscopic world holds enormous insights for the future of medicine and for your practice.